Hello students, this is the fifth part of phylum Porifera. We will study the life history of poriferans, that is the reproduction. There are two types of reproduction found in Porifera, the sexual reproduction and asexual reproduction. Sexual reproduction, as you can see in this diagram, it consists of the formation of sperm and the egg, and then fertilization. The fertilization is internal. It is occurring inside the body of sponge. So that's why the fertilization is internal. This is a question which is asked in exams. And then after fertilization, there are some sponges having an larval stage also. We will see the types of larva, and this is how the life cycle is completed. And the asexual reproduction consists of budding. So we will see how reproduction occurs in Porifera. So reproduction is of two basic types, asexual and sexual in poriferans. Asexual reproduction occurs by external budding, internal budding. This is a very important concept which is found in sponges, internal budding or gemulation, and then fragmentation. Now asexual buds, that is the external budding, so may break off or remain attached to form colonies. So the birds, they may detach from the organism and form a new organism, or they may remain attached to the main colony, to the main organism, and when many birds remain attached to the main organism, a colony is formed. Freshwater and few marine forms produce internal buds which are called as gemmules, and the process of reproduction by internal budding is called as gemulation. These are dormant masses of encapsulated cells, and they are usually produced during harsh conditions. So this is very important. This is a question which is asked, that gemulation occurs when? There are three modes of asexual reproduction. So when will the sponge prefer formation of gemmules? So it will produce gemulation during harsh condition. So these gemmules may be retained inside the parent sponge, or as the original sponge disintegrates during the harsh condition, they fall to the bottom of the water body. Asexual reproduction by budding. See, this is the main organism. At the base, an outgrowth starts growing. This outgrowth or the bud continues to grow, and the bud finally may remain attached to the parent to form a colony, or it may detach from the main parent and form a new individual. Now in fission or fragmentation, see, sponge is split or broken into pieces, which is usually due to accident. So due to an accident, the sponge body disintegrates into pieces, or it may purposefully disintegrate by fission. So lines of weakness grow, and then the small parts of the sponge separate from the body. These parts, which are splitted or thrown off from the parental body, attach to the substratum and form a new individual. So new individual attaches to the substratum and begins to grow. So in budding, a new bud forms. In fragmentation, the sponge disintegrates into many small parts, and each small part then forms a new individual. Now, sexual reproduction. Some sponges are monoecious, some are dioecious, but mostly they are hermaphrodites. Mostly they are hermaphrodites with internal fertilization. Sponges are usually both male and female, but rarely self-fertilized. That is, the fertilization is cross type. It is not self, and the fertilization is internal. So type of fertilization we have to remember, it is internal, that is occurs inside the sponge body, and it is not self, it is cross fertilization. Specialized sex cells in mesenchyme form egg or sperm. Sperms are released into the water. Eggs are retained in mesenchyme. So see, sperms, a sponge produces both sperms and ova. Now the sperms are released into the water, into the spongocoel, and from spongocoel they are released into the water through osculum. So, and then eggs are retained in the mesenchyme. Eggs, they are present in the mesohyl layer. Sperms are drawn into the female sponge through ostia or pores and fertilize the egg. So this is how sperms from another sponge are drawn into through the ostia into another sponge through ostia or pores, and they fertilize the egg. Most sponges are viviparous. See, this is not true vivipary because placenta is not formed. So they are also called as ovoviviparous. So they retain and nourish the embryo. Free swimming ciliated larvae hatch and are released. Larval forms we will see later. So sexual reproduction, we have to remember sponges are hermaphrodite, but the sponge which releases sperm is called as the male, and the sponge which takes the sperm through ostia is called as the female. The larvae are free swimming, are released, and these drift until they attach to a rock and grow into an adult sponge. Now, if you see the sexual reproduction, these are two sponges. Sperms are produced by choanocytes. They are released into the water. They enter through the ostia. When they enter through the ostia, in the mesoglia is present the ova. Eggs are formed by archaeocytes. These are the questions which you have to remember. Sperms are produced by choanocytes. Eggs are formed by archaeocytes. Sperm and egg meet into the mesohyl layer. So that's why this is an internal fertilization example. And then development starts inside the body and the larvae are released. So that's why they are also termed as ovoviviparous, because some part of the growth is occurring inside the body. And the larva, then it attaches to the substratum and forms a new sponge. The larva of sponges, they are very important. We have to remember the name or in which organism which larva is formed. So parenchyma or stereogastrula larva and amphiblastula. These are two types of larvae. And parenchyma or stereogastrula is found in the life cycle of Leucosolia. And amphiblastula larva is a typical free swimming larva found in the cycon sponge reproduction. So we have to remember in the life cycle of cycon, amphiblastula larva, in the life cycle of Leucosolia, parenchyma larva, which is a type of stereogastrula. Now see, amphiblastula, this is the diagram of amphiblastula. It is oval in shape. Now if you see, it has flagella on one side and another side has no flagella. The side which has no flagella has large cells called as macromeres. The side of amphiblastula which has no flagella has large cells called macromeres, and the side which has flagella has small cells called as micromeres. So large cells macromeres and small cell micromeres, and the small cells have flagella. So it shows small micromeres with flagella on one side. The other half of the larva shows large macromeres. The larva comes out of the sponge through osculum. It swims in the water for some time with the help of flagella. So this is amphiblastula larva found in the life cycle of cycon. Now see the parenchyma larva. Parenchyma larva is found in the life cycle of Leucosolia. This larva shows an outer layer of flagellated cells and a mass of internal amoeboid cells. So outer layer of flagellated cells is present and a mass of internal amoeboid cells. At the posterior and the non-flagellated cells are present called archaeocytes. The posterior side, which is non-flagellated, has cells called archaeocytes. This larva swims in the water for some time, then attaches to a suitable substrate. So this is about sexual reproduction in Porifera. And so we have two types of reproduction, just to revise. we will go back and see the types of reproduction. So asexual reproduction in Porifera occurs by external budding and internal buds. So internal buds are called as gemulation, and it also occurs by fragmentation, which is also called as fission. And sexual reproduction is the sponges are hermaphrodites mostly, and fertilization is internal, and it is never self-fertilization, it is cross fertilization. And larval stages, so development is direct or indirect. Development is indirect with the presence of larva, which is called as the parenchymal larva. It is found in the life cycle of Leucosolia, and amphiblastular larva, which is found in the life cycle of cycle. Thank you.